LIJO John, medical oncologist, Texas Oncology, Longview, USA. I invite Mrs. Phoebe K. John, Assistant Professor, Department of Biotechnology, to introduce and invite the resource person. And please.
Thank you, Adul. It's my great pleasure to introduce our guest of honor and speaker of today's section, Dr. LIJO John, oncologist in Longview, Texas, USA. Dr. John is a cancer and hematology specialist who is one of the physician partners at Texas Oncology Physician Associates and is affiliated with Good Shepherd Medical Center in Longview and Longview Regional Medical Center. He graduated from Govt Medical College, Thiruvananthapuram, and had briefly worked at Govt Medical College, Kottayam in internal medicine and then at Lakeshore Hospital, Kochi. He has done residency in internal medicine and fellowship in oncology at Baylor University Medical Center, Dallas, USA. Dr. John has been practicing for over 18 years since getting his license to practice in Texas. To mention other achievements in Dr. John's career, he is also board certified in the American Board of Internal Medicine and Medical Oncology. Dr. John believes that a patient and a honest discussion forms the cornerstone for the best care. He and his team strive for excellence, guiding the patient and family through the diagnosis, treatment, and follow-up. Today, Dr. LIJO John is with us to deliver a talk on molecular biology in oncology. On behalf of Biotechnology Department, IQAC, and management of Sree College, Kalady, with great pleasure, I take this opportunity to invite Dr. LIJO John to this session. Over to you, sir.
Thank you so much. All right, dear friends. It's a webinar again, and we can't seem to get enough of it lately. But anyways, I'm sitting here where it is probably close to 5 degrees Celsius and halfway across the world. I'm sure it is a warm and beautiful Saturday morning in Kerala. You're stuck with me with this super interesting topic now. So let me introduce myself. You can hear me well, right?
Yeah. Yes, sir.
Okay. So I'm LIJO John. I'm a medical oncologist or cancer specialist. My dear friend Sujaya asked me one day, can you present the topic for my students? And I said I would be delighted, but I'm afraid it may be better if they can teach me something because I am not sure how or what I can teach them. But anyway, I said I'll take the challenge, but I want to be clear what I am and what I'm not. I'm a doctor. I see patients. I'm not a research scientist, and I put patients in clinical trials, but I do not design the drugs that I use. I know the basics of the basic sciences, but sometimes even the basic sciences are quite advanced these days for me. Anyway, I was thinking of how I can be of use to you folks who are learning to be scientists and probably leaders in the biotech industry in the future. I thought maybe I could tell you a few things that I use almost every day in my work life, which is really rooted in molecular biology. Maybe it will pique your interest in the subject, or maybe it will help you see from a different perspective. So on to the topic, molecular biology and oncology. It does sound very cutting edge, doesn't it? But think about it. We're living at the cutting edge of technology, cutting edge of medicine, of oncology, way more advanced than what this world has seen in the last 5.5 billion years of its existence. Thanks to the thinking human, to the advances in genetics, in immunology, the molecular biology, you're going to be leading the lantern in a few years from now. Not a single day in my work life passes without utilizing the advances the biotech industry has delivered. I thank the wonderful human beings who are the thought leaders driving the molecular biology forward every day. So what I would like to show you now It can be quite disturbing for many people, but I still would like to do that because I want you to see the unfortunate changes in biology at a macroscopic level, not at a molecular level for now. The difficulty of the problem, the suffering that we're hoping to relieve through the science. And for that, you have to really dive deep into the molecular machinery that went wrong at some point during the cell cycle. You cannot cut these things out and expect to be cured. This is a lung cancer. That's an open section through the lungs with small dots representing spread out lung cancer. This is a locally advanced breast cancer. This is a type of a cancer called cutaneous T-cell lymphoma. Again, the same thing here. You can't cut these things out and expect to be cured. I mean, you can't keep on cutting. How much can you cut out of the body, right? You have to really know the basics of what's going on here. Probably you have to use a knife that works at a genetic level to the scale of purines and pyrimidines. So when do you think we call a condition cancerous or leukemic? Loosely, leukemia is a cancer of blood cells, but whether it's cancerous or leukemic, an initial trigger, it could be a random accidental mutation in tumor suppressor genes. It could be a mutation that happen and accumulate trying to adapt to a stressful stimuli, maybe smoking or alcohol, but whatever it is, I mean, or it could be alterations through epigenetic mechanisms. We'll go through that in a bit. Maybe a viral agent like a human papilloma virus. Whatever be the triggering stimuli, it fires up cellular multiplication. Eventually, the cells, they lose its own ability, its own braking systems for controlling the proliferation, and the collective growth, it eventually is not able to be controlled by the usual brakes of the whole organism, which in most situations it is usually the immune system, and eventually they invade the neighboring tissues or distant tissues. To understand the cancer well, you have to go back to the very basics of the cell replication industry that is happening inside our cells. I don't think I'll have to dive in detail about what's, about that with you, because you probably are well versed about these, and also for lack of time, but a quick refresher could be helpful. But please feel free to interrupt me if I'm going through things which you're probably already experts at. On the top right, you see a nucleus with the 23 pairs of chromosomes. In a human cells, packed nicely, one pair from each parent. And when you unpack and loosen the chromosome, you get this double helix strand. And you know all about the 4 nitrogen bases that form the alphabets of the sentence that ultimately dictates your fate, whether you like it or not. Well, if you don't really like it, maybe because of an unfortunate mutation, there may be ways you can edit those alphabets to your liking. Now, I don't mean to start a debate about what's ethically right or wrong, but how do you do that? I don't know, but I'm sure it probably will be coming soon in the future. So as you see, these purines and pyrimidines, they're held pretty loosely with hydrogen bonds. And so it doesn't lose and splatter inside the nucleus, not that tight and not that loose. So, it doesn't splatter inside the nucleus, and it is not that tight either. So it doesn't, separate when the time to separate comes, like during cell division. Very nice, right? So how do you pack these genes inside the cell? So this DNA strand, the blue DNA strand that you see, that's about 4 meters of DNA, 4 meters in every nucleated cell. Can you count how many cells you have? Probably billions, right? And how do you pack all that in your modest, less than 2 meter frame? Incredible packing. 1:400,000. That's the ratio of compression that goes there. If you try to fold a paper in half, and if you try to fold it again, you probably can fold it 7 or 8 times. And that's, 1:128 compression. This is 1:400,000 compression that goes on there. So that is the ratio of compression you have to achieve there. Now, how do you do that? And you probably already know this. This DNA is wound around a histone, which forms the nucleosome. And then it is coiled again, called a solenoid. And when it is coiled like that, it forms a 30 nanometer fiber, the chromatin. This is an electron micrograph of the chromatin. And this is called heterochromatin. It's thick. It is not actively transcribing because it's all wound in a very tight form. So if your cell needs to read your DNA in order to synthesize a protein, it has to be unwound first. So here, this is a very coiled form, but if it's unwound, then your cellular RNA polymerases, which is what transcribes the DNA into a protein, into an mRNA, it can read the DNA. So it has to be unwound first. And this, what you see here, is a DNA that is relaxed enough that the RNA polymerases can finally start to read it. So this is called, you probably know, this is a heterochromatin, which is a dense chromosome. And this is a euchromatin, where the DNA is ready to be read. And at this next figure, it basically gives you an idea of the level of the compressions. So this is the double helix there, and then this is the histone, and the DNA is coiled around it, and then once it's coiled again, it's as a beads on a string pattern, that's what's called. But these are the genes under active transcription. They can be transcribed to an mRNA. And this is more compacted form, less active genes, 30 nanometer fiber that we talked about before, and then you add some scaffold proteins to it. It becomes like this, and more compacted, more scaffold proteins, more compacted. And what you see here, let's go back. So what you see here is the metaphase chromosome, which is the chromosome that is trying to divide into a daughter cell. So these two chromosomes, this is the divided form, and it's held together by a centromere. So why do I go through this? What is the relation to the topic we're on, cancer and molecular biology? Well, it will come soon in the next few slides. But remember, every little detail in molecular biology, it is important, useful in some way or other. Any of these molecular processes can be altered, causing a pathological outcome, and many of these can be used as a target for developing medicine if we can master the alchemy of these tiny fibers. So let's go to this slide, which talks about epigenetics. Now, epigenetics, this is a fairly dense slide. Let me try to break it down here. We're going to talk about epigenetics, and you probably already know epigenetics deals with changes in gene activity that happens through modifications on top of the genes. And I'll show you a few example modifications. Now, these modifications do not alter the gene itself, and it just modifies the way it is read. So, although it does not modify the gene, the epigenetic modifications can have enough impact. It could potentially alter, it can have like a life-altering consequence on the organism. because it will completely affect how the genes are read. I mean, how else do you think your muscle cell behaves completely different from your nerve cell or a kidney cell or a white blood cell? They all have the same set of genes. They all came from one zygote. They ultimately led to have different fates in your body. Now, some of these modifications may be triggered by environmental factors, and some of them may be due to your genes were programmed to do that. Environmental factors, for example, like what you eat, what you drink, what you smoke, your physical activity, these modifications can be added or taken out by specific enzyme. Well, let's take a look at the slide. Let's see, on the bottom right here, what you see here is a metaphase chromosome. And as you trace it up like that, it shows more and more finer structures. And this is a completely loose DNA. And what you see on inset A is basically the histone complexes. And inset B here shows the first DNA modification we're going to talk about, the DNA methylation. Now, there's a group of proteins called DNA methyltransferases, which adds a methyl group to the nitrogen base. When you add a methyl group to the nitrogen base, it will block the RNA polymerase. Now, what did the RNA polymerase do? The RNA polymerase reads the DNA and make an mRNA out of it. And when you add a methyl group, it blocks the RNA polymerase from reading further down the strand. So methylation typically turns off a gene because it blocks the RNA polymerase activity. And the opposite, which is demethylation, it turns it on. But however, there are different situations, like for example, certain situations where if you methylate a gene, it can be turned on as well, but not all the time it is turned off. But in general, most genes are turned off when you methylate the gene. And these changes are highly regulated in the cell, and it can happen as part of normal cellular functioning or in disease processes, including cancer. For example, as you grow older, the DNA methylation decreases. Newborns have the highest level of DNA methylation. Pregnant women who were exposed to a variety of environmental adverse factors, they can change the epigenetics of the fetus, which can increase risk for schizophrenia, type 2 diabetes, heart disease for the fetus several decades down the road. And that's actually based on a study called the Dutch Hunger Winter Famine. In 1944, there was a period when mothers who were exposed to heavy famine and their kids who were later down the road, they were noted to have a higher level of schizophrenia or diabetes mellitus, and apparently their DNA methylation status was different from mothers who were not exposed to the famine. Now, heavy smokers, they tend to have less DNA methylation in certain parts of certain genes like AHRR genes, and it tends to gradually increase to the non-smoker level once they quit smoking. If the methylation happens in a tumor suppressor gene, and what is this tumor suppressor gene? We mentioned about it one time before. You probably know, but there are a bunch of genes that are responsible for preventing cells from becoming cancerous and damage the organism as a whole. Their products can do a variety of things, like proofreading the DNA when the cell divides to make sure that the 3 billion base pairs, they represent an accurate copy of the parent. And if it does not, slow down the cell division, repair the DNA mistakes, and if a repair cannot be done, start the apoptosis program to initiate cell death sequence. Apoptosis is a programmed cell death. If there is a damage to the DNA, apoptosis program will gradually start the process of suicide of the cell, so it doesn't pose a damage to others, to the neighboring cells, or become cancerous. So there are a number of tumor suppressor genes, and there's one called P53 gene, which is mutated in half of all the cancers. There's RB gene, which is mutated in retinoblastoma cancer. There's APC gene, which is mutated in colon cancer. Now, these are all examples of tumor suppressor genes, genes that are responsible for maintaining the integrity of the DNA, and if it is not able to maintain the integrity of the DNA, start the cell suicide. Another example is BRCA gene. BRCA gene is mutated in breast and ovarian cancer. If you happen to have inherited the BRCA gene, about 70% of patients, 70% of people with BRCA gene, they will have breast cancer by the age of 80. So, these tumor suppressor genes, they can be classified into three mainly: caretaker genes, which maintain the integrity or the stability of the genes by DNA repair. And then there's gatekeeper genes, which stop the cell division. They stop cell division if they detect an error. And they induce the apoptosis if there is a critical error which cannot be repaired. And then there are landscaper genes, which induce apoptosis. Landscaper genes, which they promote a microenvironment. If it is mutated, they promote a microenvironment around the cell by release of certain chemicals, which promotes unregulated cell proliferation. So tumor suppressor genes are very important in cancer biology. And we'll see more examples of how we use it later down. Now back to DNA methylation. If the methylation happens in a tumor suppressor gene, like for example, BRCA1 gene, it's basically inactivated, and it can trigger formation of cancer or carcinogenesis because the BRCA1 product is no longer made, and so there is no longer the gatekeeping functionality happening. So on the other hand, if demethylation happens in proto-oncogenes, that is, proto-oncogenes are the exact opposite of tumor suppressor genes. Proto-oncogenes, if they are activated, they induce cell proliferation. On the other hand, tumor suppressor genes, if they are activated, they stop cell proliferation. So if proto-oncogenes are activated because there is no DNA because of DNA hypomethylation, it causes cancer. Now, another example of how useful this knowledge in diagnosing is in diagnosing cancer. So there is a test that I use probably at least once a week, or maybe more than that, in our clinic. It's a test called Cologuard assay. Cologuard assay is a stools test. It's a fecal test. It is used to screen for colon cancer using fecal material without doing a colonoscopy. Easier to do than colonoscopy. It's probably not as good as colonoscopy, but the accuracy, the sensitivity is close to 90%. And it uses 3 different biomarkers to detect colon cancer or advanced adenoma. One of the biomarker it tracks is DNA methylation of a gene called NBRG4 and another gene called BMP3 gene. Now, if we're able to track that in the stool sample, if we're able to check whether the DNA for NBRG4 and BMP3 is methylated, it is a fairly reliable indicator that you may have a colon cancer or an advanced adenoma. Now, they also, that test also uses 2 other markers, but this is one of the main markers that they use. So now these DNA methylation can be potential targets for treating cancer as well. I'm going to forward the slide here, and let's take a look at this slide. So You probably have heard about acute leukemia. Has anybody heard about myelodysplastic syndrome, or also known as MDS? MDS is a very underdiagnosed disease that mostly it happens in elderly, and it is actually a clonal abnormality that happens in the bone marrow stem cells. It initially causes decrease in the white blood cells, red blood cells, and or platelets in the blood. It eventually leads on to acute myeloid leukemia, but the pace can be several decades to a few months. Some patients with MDS, they rapidly progress to acute leukemia within 6 months, but some progress over several years and sometimes decades. So anyway, what I was trying to say was abnormal DNA methylation is the main epigenetic modification that happens, and also the most studied epigenetic modification that we see in MDS. So how can we use this knowledge? So there are there are several drugs that target the DNA methylation, but there are three that we mainly use, or the most effective drugs for treatment of this condition. There's decitabine, and there's azacitidine, which is represented as AZA in the slide. And now there is another one called decitabine cedazuridine, or also called INCOVI. All of them works by interfering with the DNA methylation. Remember, DNA methylation is one of the most prominent epigenetic modification that happens in MDS. So I have probably at least one or more patients on one of these drugs who are getting one of these drugs almost every week. And here's how the azacitidine works, and decitabine works similarly. On this cartoon, the azacitidine is represented as AZA. It is basically an analog of cytidine, which is one of the 4 nitrogen bases. And it's present in DNA and RNA, the cytidine. Now, the diseased stem cell has a DNA that is rich in methylated DNA. Methylated DNA near tumor suppressor areas. So when you give azacitidine at low doses, it basically incorporates into the DNA. And when the DNA methyltransferase come to methylate the DNA, it cannot do that when the azacitidine is there. And it basically will result in degradation of a trapped DNMT. The DNA methyltransferase will get bound to the azacitidine and it will be trapped there. And that change in the DNA will trigger a suicide program or apoptosis, and eventually it will cause damage or destruction of the damaged clone. So, by the way, apoptosis, I don't know if you all are familiar with it, but it basically is, a suicide of the cell that is initiated if there's critical error. It basically has a program to cause death of the cell through a specific program inbuilt into the cellular genome. So decitabine works similar to azacitidine. It binds just to the DNA because it is a deoxy analog. And the decitabine is given intravenously and azacitidine is given subcutaneously. If I give the decitabine orally, it will be damaged by an enzyme called cytosine deaminase and it will be inactivated. What if we can inhibit the cytosine deaminase with another drug? Then we can potentially give the decitabine orally. Well, that's exactly what's done with the newest drug in this group called INCOVI. So let's take a look here. Now, when we give decitabine or azacitidine for these patients, they have to come to the clinic 5 days a week. And that's very inconvenient. But what if we can give a pill that will work similar to that? So this is the decitabine molecule, and when you ingest it, this enzyme called cytosine deaminase, it will break down the decitabine to an inactive metabolite, and it won't be able to work on the DNA methyltransferase. I mean, most of the things that you eat, like proteins, for example, it will be broken down. It will be broken down by the digesting enzymes in your gut or in your liver, and so it will be useless. Hormones, we can't usually give most hormones except thyroid hormone because it will be digested by the enzyme. But this is a molecule, cytosine, which can specifically inhibit cytosine deaminase, and so the decitabine can cross over and work in the bone marrow cell, which is damaged, and block the DNA methyltransferase. So that's very convenient, right? I mean, the patient can take pills for the same effect. I have a patient who has had excellent control of the MDS for probably a little over a year, and she's about to undergo stem cell transplantation as the next step. And she's one of the first patients in East Texas. As soon as we got the medicine approved, she started the medicine. Now let's take a look at inset C. And that's where we talk about histone acetylation, which is the next epigenetic modification we're going to talk about. It adds acetyl group to histone lysine tail, and the deacetylation process will remove it. The acetylation is done by histone acetyltransferase, also called as HAT, and the reverse process is done by histone deacetylase, or HDAC. Why is this important? Let's take a look here. So in this slide, what you see is a closed chromatin structure with the DNA wrapped around the histone quite tightly, and it is not, the RNA polymerases cannot read, they cannot use the DNA for transcribing an mRNA because it's tightly wound. Once the HAT is activated, it is going to add an acetyl group to the histone tail. And when it adds an acetyl group to the lysine and to the histone lysine tail, it will neutralize the lysine positive charge. And once the positive charge is neutralized, it will not be able to interact that tightly with the negatively charged DNA. And so it becomes loose, the DNA becomes looser from the grip of the histone. Basically loosen up and relax the DNA, and the DNA is open to be read now. And HDAC will do the reverse and close up the DNA. Now, being able to open up the DNA and close the DNA is powerful. If we're able to selectively open up just the needed areas, even more powerful. So naturally, there's a lot of effort going on in this direction. There are several medicines that work by blocking this pathway. Voronostat, Belinostat, Penobinostat, Givinostat, but I'm going to focus just on the oncologic uses for this pathway. I'll tell you that the current drugs available blocking this pathway has not yet been able to produce like a blockbuster drug for cancer, maybe because of the evolving understanding, and we may see future developments in more selective blockade, a combination with other therapeutic agents to maybe beef up the response of the treatment. This is Voronostat. Voronostat is an example HDAC inhibitor. It works by opening the DNA before the tumor suppressor genes. So Voronostat will block the HDAC and in front of the tumor suppressor genes, and it will basically activate the tumor suppressor gene because that strand of the DNA is opened up more when the HDAC is inhibited. So it is used in cutaneous T-cell lymphoma. This is a slide that shows the ultimate end effect of opening up the tumor suppressor gene, which is basically blocking proliferation, blocking migration of the cancer cell, stopping the cell cycle, initiating apoptosis, and all that. So this Voronostat is used in cutaneous T-cell lymphoma. Remember that slide from from before? This is an example of T-cell lymphoma. It is approved to be used in T-cell lymphoma. And then another example, which works by the same pathway, is romidepsin. Another example is Penobinostat, and it is used in treatment of cancer involving plasma cells called multiple myeloma. It actually was recently withdrawn from the market because the drug showed promise initially, but the company couldn't complete the trials that were needed for the final approval. So anyway, this was withdrawn from the market. This is an overview slide. Just wanted to summarize the ideas here. So in a normal cell, if this is a tumor suppressor gene here, the promoter region, which enhances the reading of that gene or enhances the transcription of that gene, it is usually acetylated and unmethylated there. So it is usually acetylated here. The histone tails are acetylated, so it's open for reading. And the promoter genes, this area is unmethylated, so it is not inactive. And so the tumor suppressor gene function goes on normally. And what's beyond here is methylated, so that's inactive. But in a cancer cell, the opposite happens. It is deacetylated, and so it's closed structure, and the promoter region is methylated. So it's the reading of the tumor suppressor gene does not happen. So let's start with the epigenetics. I'm going to switch gears to a different topic, or else I'll be accused of this now, because epigenetics can sometimes be very, very boring. But keep in mind, it's a very useful area of research. All right, let's start about something different. Has anybody heard about Philadelphia chromosome? It's one of the exciting moments in modern medicine is when a new drug called Imatinib was approved for treatment of patients with a type of leukemia called chronic myeloid leukemia, or CML. I mean, it literally overturned the fate of patients with CML overnight. That's a bit of an exaggeration, but you get it. The work that ultimately led to the development of Imatinib, it spent several years. And it is one of the most exciting chapters in medicine, where the knowledge of pathogenesis of the disease triggered a purposeful search for a molecule that will inhibit exactly where we want to inhibit, as opposed to cytotoxic chemotherapy used for this disease prior to that. And cytotoxic chemotherapy that we used to give for CML patients, it basically integrates into the whole DNA and damages the replication machinery. But this is a very targeted agent. Now let's try to understand the molecular biology there. So this is normal chromosome 9, with the blue area representing ABL1 gene. So this is ABL1 gene here, and this is BCR gene here. We don't know what this normally does, the ABL1 and the BCR. we don't know the normal function of this gene. If you ask whether don't we have a knockout mice model with BCR or ABL1, we do have knocked out mice models of mice models that have been knocked out of ABL1 gene, but all they show is poor survival once they are born. And why, nobody knows for sure. They just die. So while we don't know the actual function for ABL and BCR, this is what happens in CML, or chronic myeloid leukemia. So this chromosome 9 breaks off at ABL1 gene, and chromosome 22, it breaks off at BCR locus. And it forms a new fused chromosome. This portion fuses to the chromosome 9, and this portion fuses to this one. And so this new gene, where this is translocated, the 9 and 22, that is formed with a new fusion gene here, BCR-ABL. So now, depending on where this break happens, I told you this is the BCR portion, depending on where the break happens, we can have different, mainly 3 different formats. There's a P210 version, which is the most common form in CML, and in about 1/3 patients of a more acute type of leukemia called acute lymphoblastic leukemia. Philadelphia chromosome positive acute lymphoblastic leukemia. And then there is another form. This is basically the exons of ABL1 and exons of BCR. And P210 happens when the break happens somewhere in the middle, let's say. And this is the most common form in CML. This P190, and that's the most common form in acute lymphoblastic leukemia. And there is another one called P230, which is usually seen in chronic neutrophilic leukemia. And these have different levels of ABL1 kinase activity. Now, what is a kinase? Let's see if I have. Okay. So let me explain a little bit about what kinase is. So this BCR and ABL, when it fuses together, ABL1 we know normally has a kinase activity, and the kinase activity is adding a phosphate group to a protein in order to activate that protein. And that activation may actually cause impacts down the line. It may cause cellular proliferation or different responses. So the ABL1 kinase activity is greatly increased by the BCR just above this. And depending on this locus where it breaks off, the kinase activity is different. The P190 has the highest kinase activity, and in acute lymphoblastic leukemia, they do multiply faster. So depending on the pace of the proliferation of the disease, the CML can be chronic phase or accelerated or blast crisis. But basically, as they proliferate more and more, they accumulate more mutations than just BCR-ABL1. Because BCR-ABL1 is not, that's not the only one in the driver's seat as they multiply more and more. And that's when we call it a blast crisis. It's hard to treat that. But before that, we can treat. Now, this is the crystallographic structure of the tyrosine kinase, and it's complexed with the imatinib molecule. Now, this is the product of the abnormal fusion. And what the imatinib does is it basically latches onto the active domain of that product. So let me show you here. So let's say this is the abnormal BCR-ABL product, and it has a kinase domain, which basically adds a phosphate group to a protein that latches onto it. Let's say this is a protein that goes into that kinase domain, and it adds a phosphate group, and it's activated. And if I give Imatinib, what it does is it goes into that kinase domain here, and the substrate cannot enter the kinase site. And so it will not be phosphorylated, and it will not be causing the effects down the stream. Well, the science is robust, right? It's designed for a purpose, and it's blocking the exact, the damaged pathway. The preclinical studies looked promising. The clinical trials, they gave jaw-dropping results. The responses in the chronic phase CMO was unprecedented. Extremely sick people just waiting for the death, transfusion after transfusions. No real hope until then. They just walked back into their normal life. The approval came in a record time. So molecular biology, folks, molecular biology. What your learning and efforts can do is priceless. Today, we have second generation TKIs, second generation ABL1 TKIs instead of Imatinib called Nilotinib, Dasatinib, and Bosutinib, which achieves faster and deeper response than Imatinib. And also third generation TKI like Ponatinib, which is used for resistant CMO or for patients intolerant to other TKIs. So as we started understanding more and more of the molecular mechanisms of the cancer, and once intranet and widespread access to information is available, there came an explosion in the wealth of the knowledge. Each drug being developed is not just the fruit of one company or one group. It is the collective effort of everyone before them, with them, who were willing to share the knowledge to the whole world. So we as a human race can build more on that. And we started understanding what a driver mutation is. Now, we talked a little bit about that particular mutation for CML. A driver mutation is a single mutation that is solely responsible for cancer growth. And so started the era of precision medicine or personalized medicine. The idea is if an individual's cancer is driven by a specific driver mutation, we can use a molecule that can specifically inhibit the product of that mutant gene. Let's see. Now, it sounds very promising, but the truth is, not many cancers have a mutation in the driver's seat. They have many, many drivers driving in different directions all at once. This is a slide just to show the complexity of the mutations in acute myeloid leukemia. They're translocated everywhere. And it's not just one mutation driving the problem anymore. But it's not all doom and gloom. There are a number of patients with cancers with single driver mutation we can really exploit. The list is slowly starting to grow. With more and more understanding of the pathogenesis, we are able to design drugs that block the driver out of the seat. These are just example mutations that we are able to give a specific drug that target the actual mutation. I guess I don't have a lot more time here left, but let's go through quickly. So this is a PET scan. A PET scan is basically a scan of the active cancer sites. And what you see here is all lung cancer, spread out lung cancer in multiple areas. And this patient had adenocarcinoma, a type of lung cancer, with a particular mutation called EGFR. And there is a blocker for EGFR called erlotinib. And it's a pill. You just eat the pill, and this is after giving erlotinib. All the cancer is pretty much gone. This is not cancer. This is a heart, normal activity of the heart. This is normal activity of the bladder. That's normal activity of the brain. So you see how profound that driver mutation had. Now there are about 3 generations of the EGFR inhibitors after erlotinib. Erlotinib, gefitinib, they're the first generation inhibitors, and there's afatinib, and now there's osimertinib. Osimertinib is shown here. It's blocking the EGFR locus here, the mutated EGFR. It doesn't block the native EGFR. And this block will ultimately inhibit the pathway down the road from EGFR. These pathways can drive cellular proliferation. I don't have time to talk about all the individual drugs, and I think it's beyond the scope of what I'm trying to achieve here, but just know that these drugs have helped patients to spend that extra birthday or birthdays with their granddaughter or husband or mother with a quality of life that will make, hopefully, lifelong memories for them. I just want to spend a quick minute about next-gen sequencing as well. And it has been a vital toolkit in an oncologist box nowadays. It is used in almost everybody with a metastatic solid tumor or other hematologic malignancies to profile their tumor DNA, to find which driver mutation they may have or what drugs we could or may not use, or to understand how aggressive their malignancy is likely going to be. Now, as you know, DNA sequencing, it used to take massive amounts of time. The first human genome sequencing 3 billion base pairs, it took over 10 years. Now, same task can be done in 1 day. 1 day. And how about the cost? It used to be millions and millions of dollars. Now it's $1,000, give or take. You can have I'm sure soon it will be down to maybe $100 or something like that. You can have a copy of your entire genome in your flash drive for $400. Is your genome useful to carry on like that? Maybe. You should ask your teacher about that. Now, the cool thing is we don't always need the solid tissue from the cancer to sequence the genome, to sequence a cancer genome. We can just draw a blood sample and extract the circulating tumor cells and the DNA from that. There are a number of commercially available tests that I use almost every day for my patients, using either solid tumor or liquid biopsy technique from blood draw, Caris Next-Gen Sequencing, NeoGenomics Foundation one. And now, Texas Oncology was a huge part for a massive international trial that was designed to explore feasibility of diagnosing cancer early on using just blood tests, which can be done every year, just like cholesterol tests or something like that. And the trial is currently in the second phase. The initial results are promising. I'm sure in a few years it'll be ready for prime time. Now, I'm not sure if I have enough time to talk about immunotherapy here, but I just wanted to show you something real quick. Immunotherapy, in a nutshell, some of you may have heard about it. It just revolutionized the landscape of current cancer treatment. We know that we knew for years that our immune system is able to protect us from cancer cells to an extent. You and I may have a few cancer cells that are floating around, and our immune system fortunately takes care of them for the most part and prevent them from taking over. So patients who have been on immunity suppressing medications, for example, they're at increased risk for certain types of cancer like lymphomas and skin cancer. So the question was, how can we unlock our immune system and direct it against cancer cells when and where we want it? That's a cancer cell, this large spiky cell. And your immune cells, these small ones, the macrophages, they're basically injecting some toxins into the cancer cell and trying to destroy it. Now, some very basic concept. We have an innate immune system, which is inbuilt or encoded to you. You don't need a vaccine or exposure to an organism to learn. And then we have adaptive immunity. For that, you adapt as you grow. That's where we use vaccines and all that. And the innate immunity has different components. It can be a macrophage cell like that, or a neutrophil cell, or a natural killer cell, etc. And the natural killer cells are somewhat unique in that they don't directly kill the pathogens. They kill the compromised host cell. If a host cell is infected by a virus, or whether the host cell has become cancerous, then they damage the host cell directly. So they work with what we call as self-antigens. By the way, an antigen is basically a protein or a structure that is capable of inducing an immune response, and an antibody is a protein that is released by your immune cells, which can latch onto an antigen. Now, by attaching to an antigen, it will trigger an immune response cascade through various mechanisms back to natural killer cells. So we have a set of cell surface proteins called major histocompatibility complex. And MHC, or major histocompatibility complex, is what teaches your immune cells that, hey, this cell belongs to self. This is mine. Leave it alone and don't attack that cell. The NK cell works by identifying the missing self proteins in the damaged host cell. So what do I mean by that? If your host cell is damaged by virus, or if the host cell becomes cancerous, the MHC proteins, or the major histocompatibility complex proteins, are downregulated. And when the natural killer cells, they stop and scan during its routine security checkup, and they scan the surface of the cell, they will see that the MHC is no longer expressed in needed quantities. And when it's not in enough quantity, it will basically transform to a merciless killer. Well, in real life, it probably just pours a protein called perforins and proteases, which are like bullets. They punch a hole in the cell membrane, and things will just start leaking in and out of the cell. And the cell will either start the suicide program, the apoptosis we talked about before, or it will die from osmotic lysis. Let's take a look at this slide. So the blue cell is called an antigen-presenting cell, and they process the antigens and present it to T lymphocyte. T lymphocyte is one of the immune cells that your body has, and it needs a T cell receptor to present the antigen. It's teaching the T lymphocyte. This is something that you should be triggering a response against, okay? Now, when it does that, let me just go on to the next slide, which will be probably easy to understand. So this is a T cell, this is an antigen presenting cell, and it presents an antigen through the MHC. There's a type of MHC2 proteins, and the T cell receptor recognizes that. And then when it's triggered by another stimulation, just the T cell receptor TCR interaction is just not enough to activate the T cells. T cell activation is very important for cancer recognition or fighting against microbes. Activated T cells can kill cancer cells. So T cell receptor is there, but it also needs one more stimulus. And it can be a CD28 engagement, which will stimulate it, or it can be a CTLA4 engagement, which will inhibit the T cell. So this is a stimulatory signal, and this is an inhibitory signal. What if we can block this CTLA4 with an antibody? If you do that, what's going to happen is you're blocking the inhibitory pathway. And this pathway is called checkpoint. It's an immune checkpoint. That is, if there is a antigen recognition, and if the CTLA is engaged, it stops the attack. It's an immune checkpoint. But if you can block the CTLA, you remove the inhibition, and the T cell will always remain active, thereby increasing the cancer response or attack against cancer. Does it work? Yes, it does work. And that's how a medicine called ipilimumab works. And it is used in melanoma, kidney cancer, and liver cancer. The problem is, some patients, if these T cells if they remain unchecked, it can cause a lot of damage to other tissues as well. But fortunately, we can put a brake on these effects as needed. Let's take a look at a different mechanism. This is a protein called programmed death PD-1, and this is another one called PD-L1 programmed death ligand. Now, this is a tumor cell. The tumor cell is your own cell. It learns how to evade immunity. And if the tumor cell starts expressing more of the PD-L1, and if it is able to latch onto the PD-1, it's like a flip-off switch for the T cell. And so the T cell stops attacking the tumor cell. What if we can block this PD-1 receptor with an antibody that can attach to the PD-1? And so this flip-off switch will never be activated. And so the tumor cells will be damaged by this medicine. This medicine blocks an inhibitory pathway, and so the T cells can damage the tumor cell. So does this really work? You will hear a resounding yes from people all over the world who are alive thanks to the discovery of this pathway and drugs that work by blocking this pathway. And boy, oh boy, did it unlock the jackpot. It looks like it did. If you go through the currently approved indications for pembrolizumab, which is a PD-L1 blocking agent, I mean, this is the wide variety of cancers this medicine works on. I'm going to stop the presentation there and switch over for some questions, because I think I may have really gone over the time limit that was allotted. So I'm hoping that, I was able to give you another perspective on molecular biology from a practicing doctor's viewpoint and unleash hopefully a new level of enthusiasm for my budding molecular biologists out there. But anyway, time for questions. Can you hear me all?
Yes, sir.
എന്തെങ്കിലും സംശയങ്ങളുണ്ടോ? ഇപ്പൊ സംസാരിച്ചല്ലേ, കുറേയേറെ സംസാരിച്ചു തോന്നുന്നു. എല്ലാവരും അടുത്ത് കാണുമല്ലേ?
Thank you, sir, for your very explanation. Now is for the introductory session, and I request the participants to come forward with their query in the chat box.
എല്ലാവർക്കും എല്ലാം മനസ്സിലായോ? പറഞ്ഞുകൊണ്ടിരുന്ന എല്ലാം മനസ്സിലാക്കണമെന്നില്ല, പക്ഷെ ഹോപ്പ്ഫുള്ളി ഞാൻ നിങ്ങൾക്ക് കൂടുതൽ അറിയാവുന്ന കാര്യങ്ങൾ പിന്നെയും സംസാരിച്ചില്ല എന്ന് പ്രതീക്ഷിക്കുന്നു. അത് ഇനി ഞാൻ സംസാരിച്ചത് ഒത്തിരിയേറെ കോംപ്ലിക്കേറ്റഡ് ആയിട്ടുള്ള കാര്യങ്ങളായിരുന്നു ഒന്നും എനിക്ക് അറിഞ്ഞുകൂടാ. നിങ്ങളുടെ ലെവൽ ഓഫ് നോളജ്, നിങ്ങൾ എന്താണ് സിലബസ് പഠിക്കുന്നത് എന്നുള്ള എക്സാക്ട്ലി എനിക്ക് അറിഞ്ഞുകൂടാ. പിന്നെ ഇതൊക്കെ ആയിരിക്കും ഒരു ഊഹത്തിലങ്ങ് പ്രസന്റ് ചെയ്തതാണ്.
So some questions are there on the chat box. Shall I read out?
എനിക്കത് കാണാൻ പറ്റുമോ നോട്ടെ? പറഞ്ഞോ.
Are most drivers of cancer detected in each patient before providing the drugs? Isn't it a time-consuming process, both the detection as well as the treatment of the patient?
Yeah, so when we see a patient, let's say somebody who has lung cancer, and let's say they have metastatic lung cancer, it's usually a process, if you're looking at lung cancer, it's usually a process that happened, over several months. And when we see the patient, we start the process of profiling the tumor. A solid tumor profile would take about 10 to 14 days. And, we could also use liquid biopsy these days, which gives a result within 3 to 5 days. So waiting 10 to 14 days for a process, for a cancer that took years to develop or months to develop, that's okay. You're not going to really change much by waiting 10 to 14 days. Now, in some patients, if they want to start treatment right away, we can start treatment and then modify the treatment according to the results of the molecular profile that eventually I will get within 10 to 14 days from when I see the patient. So it is time consuming, but it's not longer than generally 10 to 14 days, and it's getting faster and faster as well.
Okay, sir.
Did I answer the question?
The next question by Bhagyalakshmi. Sir, is this CTLA inhibition is a restorable process?
The CTLA inhibition using ipilimumab. So I suppose what you're asking is whether it is irreversibly blocking the CTLA4. Is that what you're trying to ask?
Yes, sir.
Okay. It is not, meaning so if you're looking at the effect of the drug, so if I give ipilimumab for a patient, it will work for, the drug will be in the system for a few days, and the effects will start slowly weaning away. It's not like, once you give the drug, the effect will keep on happening. It will fade away after a few days. So you have to repeat, you have to give the drug generally every 3 weeks or 2 weeks, depending on the dose, to keep on inhibiting the CTLA. Now, I don't know for sure whether the ipilimumab irreversibly binds to the CTLA4 or not, but the efficacy does not last too long, because even if it irreversibly binds, the life of each cell, the lymphocytes, is not long enough that it will really matter after a few days, because the lymphocyte that you inhibited will die away within a few days. So we have to keep on giving the medicine, and the lymphocytes will be constantly repopulated by the marrow, and as we give more medicine, it will block. Now, honestly, the CTLA4 pathway, we use it. It has actually combining CTLA and PD-1 gives a CTLA pathways will give a better response, but it is also dealt with more toxicities, and so many oncologists would sometimes use just the PD-1 pathway. But anyway, the effect won't last too long, to answer your question. Did that answer the question?
Yes, sir. Thank you, sir.
Okay. I see the chat box here. So can treatment with personalized drugs completely replace radiation and chemotherapy? So remember the slide where I showed you the AML mutations? So personalized drugs can work when we have, one or maybe two driver mutations. Now, when you say personalized drug, we're talking about identifying a driver mutation and using a medicine to target that. So it works when there is a cancer where there is one or two driver mutation. What if they have so many driver mutations? What if they have, like AML, like that slide that I showed you? There's just too many mutations that we can use a single drug to inhibit. So there is a role for chemotherapy still. but there is a role for driver mutation targeting in those types of cancer. Now, as we understand more and more about the biology of these cancers, like for example, the immunotherapy, for example, you don't have to know the driver mutation for the immunotherapy to work, and it works pretty well, even in cancers with driver mutation. So we may have to use a combination approaches to ultimately get a tumor cell death. But will it completely replace radiation? There's a role for radiation. In locally advanced tumors where surgery is not an option, we can still use radiation therapy in those situations. But if it's metastatic, that's where more of these targeted agents come into play, where you cannot focus in one area with radiation. There's too many widespread disease to use radiation. And in those cases, yes, we can use personalized drug. But, in the next 10 years, it will be different, I'm sure. You may 20 years down the road, an oncologist will be looking back at me and saying, what am I doing here? What type of crude treatments are you doing here? So as science evolves, I'm sure we'll be able to change treatment from surgery or radiation or chemotherapy to more and more targeted drugs. So the next question I see, how can epigenetic modifications be reversed? So from a cancer standpoint, on that, I don't know if you remember that the MDS slide where myelodysplastic syndrome clones, they have more and more of DNA methylation going on. And as we use DNA methylation inhibitors, we were able to kill those clones with methylated DNA. So in that sense, I guess it can be reversed. For reversing the epigenetic modification that was built in due to environmental factors, like, for example, somebody who smokes a lot, their certain types of gene called AHR genes may be methylated in those patients. And as time goes on, your body reverses it automatically. And as time goes on, within a few years, those methylation will automatically be taken out. So epigenetic modifications are always reversible by the body. We just don't know how to unlock, we don't know how to target one particular area and do the change, but it is always reversible by the body. It does not alter the DNA as well, by the way. Is there any, so next comment I see, is there any early detection for these myelodysplastic syndromes? So myelodysplastic syndrome is a very, very wide presentation of symptoms. What I mean is some patients may have very slow pace of the disease that when I look at their blood, initially I may not see any change for sometimes years. And eventually I will see that, okay, the red cells are growing larger in size or their platelets are dropping down slowly. And it may be a several year-long process. And in some patients, it may be a rapid process. So, early detection, we can get it done by doing bone marrow biopsies. We can check on early, indications of myelodysplastic syndrome. The problem, though, is it is not curable. Whether, as of now, whether you detect it early on or later down, there is not a known cure. We can treat it in a way that we can control the pace of the disease and decrease the rate of transformation to leukemia. But ultimately, there is no cure for these type of conditions. Now, we can do a bone marrow transplant. The problem is MDS is often seen in elderly, where a bone marrow transplant may not always be the best course of action because they are already so frail to begin with. So yes, it can be detected early on with bone marrow biopsies, but sometimes the changes in the bone marrow may be very minute, very subtle, that even if I do a bone marrow biopsy, I might miss it early on. So if I find it early on, if I see some changes and if I do a bone marrow biopsy early on, I may not be able to detect it because some of the changes are too subtle, too mild, that it will be completely missed. And then when you check it maybe a year down the road, yeah, there are the changes. But what is the benefit of finding it early? Not much in most patients.
Hey, Dr. John, this is Dr. Khan.
Hey, Rashad.
Hey, a great presentation. So as I was listening, I think I had a question that I'm not sure whether you or our colleagues who are joining us can kind of participate in. It's something that piqued my interest, and I think certainly with this being an international webinar and we've got an audience in India, I think it'd be awesome to kind of hear a perspective. What I heard was, immunotherapy is becoming so popular nowadays and it's so effective. But I've had a couple family members and relatives who've been in India getting treated for certain cancers, and the word on the street is, and I don't know how true this is, but it's been said that immunotherapy is not as effective in the Asian subcontinent and that patient population particularly. And that kind of just made me kind of interested in knowing, and it may be something our colleagues can kind of look into as they further research into this, as to seeing, the efficacy of different patient populations and different ethnicities and how they respond. And I'm just curious of whether, this Asian subcontinent has a certain limitation or resistance to these medications. It's just interesting when I heard it.
Mm-hmm. Yeah, I don't know the accurate answer for that, Rashad. I wonder if it is due to lack of selection in those patients, whether they do the PD-1 staining and all that, accurately, or whether it is due to storage of these medicines,
exactly,
the stability of the drugs, and maybe a lot of variables there at play. I'm not sure.
And I don't think I couldn't find a solidified answer, and it may be just kind of hearsay, but if there is some validity to it, I think it definitely is a field to kind of look into and explore if anybody is looking into these things, because this is like the up and coming next frontier in chemo, right? So
absolutely, yes, yeah. Harvesting the immune system to deploy against the cancer cells is definitely the upcoming thing now. And there's more and more things in immunotherapy that I haven't touched on this presentation, but it definitely is the promising approach to deploy against cancer cells. Yes.
Awesome. Thanks, man.
So I see a question. How has stem cell transplant sped up the recovery from cancer compared to chemotherapy? Ashley, I'm trying to understand the question. So do you mean that we can use stem cell transplant to speed up recovery compared to chemotherapy? I'm not sure I really understand the question, but let me tell you maybe an example which might help answer the question. So let's say you have multiple myeloma cancer, and where we use chemotherapy, and once you get a good remission from using after chemotherapy, and there are targeted agents that we can use with multiple myeloma, but once you get a good control of the disease, then you can do stem cell transplant to a type of transplant called auto transplant to basically wipe out any remaining cancer cells in the marrow. But before you do that, collect a few normal stem cells from your marrow, your own marrow, and then transfuse it back. So what you transfuse back hopefully won't have any more myeloma cells. I'm not sure how it would be possible to speed up the recovery. Ashley, can you clarify on that question? Okay. So let me go on to the next one.
Sir, can you hear me?
Yeah, I can hear you now. Yes.
What I meant was, compared to chemotherapy, using stem cells, does it help a person to recover faster? Because chemotherapy is conducted over a certain period of time, whereas stem cell transplant happens once, right? So
yeah.
Does it speed up the process of the person getting better, or does chemotherapy still have to be used?
Okay, so yeah, the chemotherapy still has to be used. One thing is stem cell transplant. Let's say, let me take an example. Acute myeloid leukemia, let's say. You cannot do stem cell, you cannot transplant somebody else's stem cells into your marrow before you kill the leukemic cells in your marrow. Because if you do that, no matter how close of a match you get, if you just transplant a stem cell into a marrow that already has leukemic cells, the leukemic cells are going to take over the marrow. So you have to first kill the leukemic cells in the marrow using chemotherapy, and we call it an induction therapy, induction treatment, okay? And that will decrease the leukemic cell, and then eventually you transplant a healthy stem cell from another donor called allogeneic stem cell transplant to the marrow, where there is literally or almost no cells remaining, not leukemic cells, not normal cells. And you just transplant that stem cell to the marrow, hoping that the stem cell will populate that new house. So you have to have a marrow with no leukemic cells. You cannot expect the stem cell to kill the cancer cell. They just don't, they cannot do that. You have to have some sort of drug or immunotherapy or chemotherapy to kill the cancer cells that is already there. And did I answer that?
So before stem cell therapy started, we just give chemotherapy and expect the cells to regenerate, as in the immune cells or the T cells. We just expect it to come back after the chemotherapy. How is it that cancer was treated back then, before the stem cell therapy?
So before the stem cell therapy, now, there is a little bit of pseudoscience going around stem cell therapy these days. Like I've heard, there is clinics, claiming stem cell will be like, the cure for everything. So stem cell therapy, I'm not sure what exactly they mean when they say the stem cell therapy. In a cancer standpoint, for you to expect the stem cells to kill the cancer cells, that just doesn't happen as of now. We cannot expect another person's stem cell or your own stem cell to kill the cancer cells. You have to have some sort of drug, whether it's targeted or immunotherapy or something like that, to kill the cancer cells, and then you can put your stem cells in the marrow. That's where you use stem cell transplant, not for solid tumors. So you only use it in a cancer involving stem cell problems like acute leukemia, myelodysplastic syndrome, multiple myeloma. Now, several decades ago, we used to use stem cell transplant for breast cancer. Now, in that, the stem cell is not the one that kills the breast cancer. What we would do in metastatic breast cancer several decades ago, you just give a heavy dose of chemotherapy and you kill the breast cancer cell and you kill your stem cells as well. And then you use your own stem cells that was stored back, infuse it back. Now that's completely different. But if you were thinking that the stem cells can kill cancer cells, I don't know of a way how it would do that. Okay. Ashley,
Thank
did
you,
I
sir.
answer you?
Yes, sir. Thank you.
Okay. So next question, is it possible to completely cure cancer? If not, will it be possible in the future? So Durga, a cure for a disease, it will come with time. So for example, I was not able to cure metastatic, let's say, metastatic melanoma 10 years back. Now I'm able to cure, not everybody, but a few patients I have had 3-4, they came with metastatic melanoma, widespread, and 3-4 years down the road, on the scans, I don't see any more evidence of the cancer. So hopefully they will never develop it. So, why am I able to do that now? Well, not for everybody, but a few patients, they get such a beautiful response. Why am I able to do that now? Because the science has advanced, and we know that melanoma is one such cancer where immunity has played a huge role. And by developing immunotherapeutic agents, at least a few patients are able to see, durable responses, remissions, and even cure. So as the science advances, I'm sure it will be, happening more and more in cancers. Another example, metastatic testicular cancers. It has a very high cure rate. Once the specific chemotherapy regimens called PEP regimen was developed, the cure rate has gone up almost in the 80s, 90%, even if it's metastatic testicular cancer. Another example, widely metastatic large cell lymphomas. We're able to cure that in more than 80% of patients. Why? Because the science has advanced. So as time goes on, as we understand more and more of the molecular pathogenesis, I'm sure we will be able to get better and better cure rate. And that's why molecular biology is important, to understand pathogenesis and to understand how to develop drugs. Did I answer your question, Durga?
Yes, sir. Thank you.
Okay. Next one, Devika. What is the difference between cytotoxicity and anti-proliferative activity with respect to cancer chemotherapeutic agents? So, chemotherapeutic agents, some of them work by directly linking to the DNA and damaging the DNA and causing what we call as direct cytotoxicity. Some of them works by decreasing the proliferation rate. For example, with CML and the Imatinib agents, eventually the cell dies as time goes on because a cell cannot live on forever. Can you clarify on what exactly you mean? Are you talking about cytotoxicity in immunology context? Because I'm trying to understand your question here. Cytotoxicity basically means damage to the cell, anti-proliferative activity, and that is basically decrease in proliferation, which is more so with more targeted agents by targeting the pathway that causes the proliferation. So I'm not exactly understanding the question there. Sorry. Devika, do you have a comment on that? What you meant? Well, any other questions, folks?
So I think almost all the queries were cleared by a speaker. Thank you, sir.
Thank you.
Now, it's wind up our session. I invite Dr. Renjisha Menon, Assistant Professor, Department of Physiology, to deliver the vote of thanks. Ma'am, please.
Thank you. It's my honor to give a vote of thanks on this occasion of our international webinar series Biotechnology, the Present and Future Scenario. On behalf of the Biotechnology Department, I would like to extend our deep obligation and hearty gratitude to our resource person, Dr. LIJO John, for his informative and interesting talk on molecular biology in oncology. Hope you all had a good experience with the session. The webinar provided broad insight for the life science students to understand the importance of molecular biology in the extensive area of cancer. The talk was very valuable for all of us. Thank you once again, sir, for sparing your valuable time with us and for the well-structured and expertized presentation. Now, I take this opportunity to express my sincere gratitude to our principal, Dr. A. Suresh, for the encouragement and constant support rendered in the success conduct of the webinar. I would also express my thanks to our management for the continuous support. Now, I would like to thank IQAC coordinator Dr. Preeti Nair for her wholehearted support to organize the event. Dr. Valsa Eki, who has always been there for us in every effort. I extend my sincere thanks to you, ma'am. After all, I would like to thank my colleagues and staff members who had worked hard for conducting this webinar. I am very happy to thank all the students and other faculty members who actively participated in the webinar and made it a great success. Once again, thank you all for making this webinar a memorable one. Thank you.
Thank you.
Thank you, ma'am. Dear participants, our today's technical session is over. We're again reminding you to fill the feedback form before leaving. Once again, I thank everyone for being here. Let's disperse for now. Thank you all. Have a nice day ahead.
Thank you.